So our next speaker is Ilja Arts from Maastricht University. She'll be talking to us today about data modeling in systems medicine. She serves as scientific director of the Maastricht Center for Systems Biology, and her research focuses on integrating large multi-omics and clinical data sets to uncover biological mechanisms behind chronic disease. So the floor is yours.
Thank you very much, also for the invitation to speak here today. We are generating more and more data. That is not a secret. That is not something I have to tell you. We've seen that today. There's so much technology, and all of that is generating more data. There are many ways to analyze these data, and much of what you've seen today is really sort of a statistical, data-driven approach to data analysis. And that is something that we in our group have been doing as well over the last couple of years, a lot. I'm just going to give you two very brief examples. This is one of them where we studied the infant gut microbiota. And here you see an iris plot of 1,000 one-month-old infants and their gut microbiota composition related to antibiotic use, indicated by the very small orange dots at the left. So you clearly see there are associations between the microbiome composition and antibiotic use. On the right hand, you see again plotted associations with these microbiota to the consumption of milk, human milk oligosaccharides. And all of this is visualized in a very nice way by PhD student David Barnett, and he also made this available as a package. A second example, which was recently published by another PhD student of ours in Nature Medicine, is where we looked at BMI discordant cardiometabolic phenotypes. So not everybody who has a high BMI is also metabolically unhealthy. And we actually zoomed in on this group, trying to identify people who have a cardiometabolic profile which is discordant with their BMI. And we found some very interesting clusters that are indeed discordant. And in fact, when we studied their disease risk in the future, so incident disease risk, we indeed noted that these profiles added something to the predictive capacity of models that indicate what your risk of future cardiometabolic disease or type 2 diabetes is. So very useful. However, what all these approaches to data analysis largely ignore is this. All the biological knowledge that we have and others have accumulated over the last decades. So, what we are doing more and more nowadays is actually trying to use that biological knowledge in our data analysis. And this started 20 years ago when the first data scientists and bioinformaticians started converting these static images that you see on the left here into interactive pathway models. These pathway models organize knowledge, they represent it in a graphical way, they are machine readable, and they give much insight into biological mechanisms underlying certain observations that we do. So Dr. Martina Sommer-Koopman is one of the architects of Wikipathways, which has been since the beginning of this decade, or a bit longer even, have been working on collecting this biological knowledge in an open source, community curated way, so that everybody can use this information to map their data, visualize their data, and analyze their data. And more recently, she did so in a large project where, together with a lot of international collaborators, COVID-19 pathway maps and subsequently disease maps were created. This is a beautiful image, and it very much resembles that static picture, but it is not. So this is a graphic that is actually interactive, clickable. You can map your data on it. You can analyze your data on it. It's an amazing data source and very useful, generating lots of insights. Only this year we got a grant from ZonneW to actually do the same for post-COVID. So we're adding on top of this, these COVID databases, we are now adding knowledge on post-COVID, in particular focusing on neuroinflammation. And we also intend to integrate a lot of different data modalities in there, including brain imaging. So these pathway and network analysis have been extremely useful. Very easy to generate pathway statistics, visualize data, very successful. However, they also have their limitations, right? They are a static snapshot of the system at one point in time. The size of a particular pathway is usually determined by the person who curated it, right? You have to decide basically what is one pathway. And sometimes you can connect that to other pathways through network analysis, for example. But by definition, they are limited. And finally, it is not trivial to conclude whether an entire pathway is up or down, so whether the sort of the net effect of up and down regulation of a number of genes in this pathway results in a net activation or deactivation of that particular pathway. And that is the reason why we are now, for a couple of years, have been trying to use so-called genome-scale metabolic models. These are mathematical models, so not necessarily very easy to visualize, but they are really built to do effective calculations throughout the entire metabolic network. They contain all metabolic reactions that occur in human bodies and all the information on genes, also the localization of the reactions, all metabolites. So they are very good at sort of estimating what happens in the entire network in a dynamic manner. They can also be used to map and visualize data, but they are particularly suited to study dynamic behavior across the entire network. So not limited by pathways that are not connected to each other. And we can, for example, ask the model to maximize ATP production. And then use transcriptomics data, for example, as a proxy for enzyme activity. And then the model comes up with an optimal combination of fluxes to achieve this. So a couple of years ago, we started working with this, and this is published work, also available as a tool, where we first mapped, analyzed, and then simulated the effects of amino acids in the human system using these gems. That was very nice work, but we very quickly realized that looking at everything, so looking at the entire network, was not a good idea. Even though they are mathematically very efficient, it was still quite a task to do this. So what we are working on now is really what we call a metabolic task analysis. And this tool was developed a couple of years ago by Rochelle. And what it does is that it splits the entire genome-scale metabolic network into smaller parts, and these are called tasks. And there are predefined tasks, but you can also develop your own tasks. And then an example of such a task could be ATP production. But you can basically think of any task that a metabolic network would have to do. So how does it work? We have been using transcriptome data, but you can use other data as well, basically as a proxy for enzyme activity. And we use these data to calculate the reaction flux through the network. We use tissue transcriptomics data. In the example that I'm going to give, we actually used heart tissue, so small biopsies from heart tissue. If the enzyme expression throughout the network is high, we get a high reaction flux. And what we then do with that is calculate an activity score throughout the network based on the individual fluxes of the reactions in that network. And then for each task, each individual gets a score, and with these scores, we can basically do whatever we want, right? So you basically get, for each individual in a cohort, for example, a score that is based on the flux through the network for all these different tasks. So we applied this tool and improved this tool to a study on dilated cardiomyopathy, where we used a public data set on non-diseased donor hearts and dilated cardiomyopathy hearts that were explanted, so end-stage disease, and the data we had were RNA sequencing. And we first validated the tool. So what you see in this figure is tasks. For example, here on the right you see fructose to glucose conversion, which is a task that you expect the network to be able to conduct, but not in all tissues, but only in specific tissues. So in particular, the liver is good at this, and other tissues not, and that is exactly what we were able to reproduce. So this demonstrated that this task analysis actually gives you credible information that fits with the biological knowledge that we have. So what we did then, and this was, by the way, work by Bastien Nihan, a PhD student, and this was recently published on bioRxiv, posted on bioRxiv. What we then did is that we used all these task scores that we had calculated for each individual and did a clustering analysis on them. And what we could see was that there were two clusters or metabotypes. Oh, sorry. And of course a control cluster that were really quite distinct and that were not picked up when we did conventional gene expression analysis. So this type of analysis really seems to add something on top of the more traditional analysis tools that we have. We then also compared the individual task activity scores between these metabotypes, and we could clearly see differences in some tasks that could indeed be meaningful for different types of patients with dilated cardiomyopathy. And one example here on the right is the sarcoplasmic calcium pump, which is known to deteriorate in dilated cardiomyopathy. And you see that these two patient clusters here on the right actually differed quite substantially, indicating that there might be distinct clinical phenotypes. With this data set, we did not have any more clinical information because it was a public data set. However, at this moment, we are repeating these analyses with a larger, better data set, again using biopsies from the heart, where we do have all these clinical information, and we hope to report back to you in the not too distant future with some really interesting results. So in conclusion, supplementing this type of analysis, so using genome-scale metabolic models on top of pathway analysis and data-driven analysis, will give you additional information without generating more data, right? It's just another way of looking at the same data. It contains a lot of detail, but there are also some issues. Anybody who has ever worked with these genome-scale metabolic models knows that they are not perfect. There are lots of issues with the code. We solved that for the task analysis. We did use the most recent version of the human genome-scale metabolic model, human 1, but there are lots of errors in there. For example, for the heart, ketone metabolism is essential. That was not present in the model that we used. So we first had to improve the biological knowledge of the model. And I think that is really where I hope we will also be able to put some community effort in. I think these models are very promising, but they really need to be curated better. Just like Wiki Pathways is a community-curated effort, I think we should be doing the same for these types of models. Two other things that we're doing at the moment is to try and improve from using transcriptomics as a proxy for enzyme activity to using protein concentrations and using AI. We are now trying to also use predicted enzyme activities. And another step that we still need to take is to go towards multi-omics. And of course we can do that by correlating these task scores to any omics data that we have. But what I think would be really nice is that we can actually look at the products of these highly active pathways and see whether we can find these in the circulation of people who have a high activity, right? So we can really start looking at what is the result or the product of such an active pathway. That can be metabolites, but it can also be proteins or protein products. So that brings me to my one but last slide. I definitely would like to thank everybody who has contributed to this work, and in particular Marianne Breuer, Michiel Adriaans, and Martina Sommer-Koetman in my lab, who are leading this type of work, and Bastien as a PhD student doing much of the work that I presented today. And then I would like to end with advertising the next BioSB conference. I'm sure that many of you know it and have probably already registered. The annual conference of the Dutch Bioinformatics and Systems Biology community will take place at the end of May in Baarlo, which is the north of Limburg, beautiful surroundings. And please, I think it's really interesting to go there for many of you. So if you haven't registered yet, please do so. Thank you.
Okay, thank you for the presentation. So, do you have any questions? Yep. Okay, I will help you.
Thank you, Ilja, for advertising this, but also for your very insightful talk. So, we have heard a lot about single-cell technologies these days. So, would it make sense to create a genome-scale metabolic model for every single cell type that we know?
Yeah, that is possible. I mean, there are, it has been applied to single tissues, I would say, or single cell types. I'm not sure we should go further and really make a model like this for every single cell, but I think in principle it is possible. So it would definitely be interesting to explore that.
Well, yeah, so my guess would be that it is very useful and that it will add a lot of context to what we're seeing, because my biggest problem is with the genome-scale metabolic models and also with the pathways is that they are so generic, so not every reaction happens in every cell. And I think we need to take that into account.
No, so there are algorithms that are able to create tissue-specific models, but they are not so very good in general. I mean, they really need work, and I think not a lot of people have been working on these models over the last couple of years. I mean, they have been around since, I don't know, 10 years, because everybody who starts working with them finds out that they are really not so good yet. And I think we have people in our group who really see the value of this and are really pushing this forward. So if anybody has good data and is interested in working together with us and see how we can make them useful, we're very happy to do that. Yeah.
Do you have more questions? Maybe one small question. So basically you're taking RNA-seq data as a proxy for the protein like intensity of functionality, etc., right? But you're basically, at its current state, you're ignoring alternative splicing, you're ignoring PTMs. Okay. Are you taking into account alternative splicing? Is it different nodes? Are you taking into account PTMs as different status of the protein? So yeah, can you talk about the limitation here a bit?
No, yeah, I mean, clearly it's a very large limitation, right? Because we are using just simple RNA-seq as a proxy for enzyme activity, and there are lots of steps in between that. But in principle, you can also use epigenetics data or any type of data. We are just using the mRNA data because these are the data that are most available, right? And we are still developing the tools. So then it's the easiest type of data to use.
In terms of mathematical models, you're probably using ordinary differential equations, right?
Sorry?
Which mathematical model are you using for your networks?
We are using a recon, no, sorry, we're using human 1.13 or something like that.
But this is basically boils down to ordinary differential equations, or?
No, these are steady state models.
Oh, okay.
Yeah.
I see. Yeah.
Yeah.
Okay, thank you. Thank you very much.